Thank you. I would like to thank you and Sophie, Lendert, and Didier for the invitation. As you say, I consider myself a friend of the group, and it's always a pleasure. As we were talking with Stefan a minute ago, it's a pity that we cannot do this in person, but still, I think you have managed to put together a very lively meeting. So lots of interesting talks and discussions. So I think that's already very nice. So thank you again. Okay, so I would like to talk, as Genevieve was saying, about embryonic development, but well, and in particular about cell-fate decision and the robustness of cell-fate decision. But maybe the summary that I would like to share with you is that I'm going to be talking about simple models applied to complicated experiments. That's basically the idea. And I can say this because we didn't do the experiments ourselves, of course. The experiments were done in the lab of Kat Hadjantonakis at Sloan Kettering by her postdoc Nestor Saiz. And from our side at UPF, Laura Mora contributed to the modeling, and she's right now doing her PhD in Bayer. So as Genevieve has already mentioned, I would like to focus on a specific type of living process or biological process in which I think physics has a lot to say. And we have already been discussing about this in this conference, and we will continue doing so today in the rest of the talks, and it's self-organization. And maybe my claim that why this fits well in this type of workshop is that the physicists have been gaining a lot of understanding about self-organization in non-living matter for the last 3 decades at least. So in principle, I think it is fair to say that we know a lot. And in particular, I would like to say that a big role in this was played by the Brussels School. So I would like to highlight this. So we know quite a bit. And of course, it was only natural that we tried to move to a very exciting example of self-organization that we can find in biological systems. There are many examples. And I would like to focus today in mammalian development, which in my opinion, as I guess you all agree with me, that it's pretty amazing. No, you go from one undifferentiated and simple cell, nondescript cell, and then in a matter of days, you can get adult organisms, in the case of mice, for instance, which are extremely well organized. And the question is, how does this happen? And of course, you can frame this question in many different ways. One way of framing the question is, okay, this organism has multiple tissues, and the relative sizes of these tissues is very robust through development, even taking into account that the natural variability of these embryos that can lead to organisms of relatively different sizes, and yet the sizes of the tissues are the relative sizes of the tissues are well controlled. So where is this robustness coming from? So we used to try to answer or to address partly this question, we used the experimental system that Kat has in her lab. And this is basically early embryonic development in the mouse. And in particular, we were focusing on the stage, on the blastocyst stage, in which the embryo has from between 50 to around 200 cells. And in this particular stage, there are a couple of cell-fate decisions. And this is closely related to what Silvia was presenting yesterday as well. So I will be referring back to what things that she already mentioned in the next few slides. And the idea is that we have a first decision in which cells differentiate into either trophoectoderm or inner cell mass, these green or purple cells that we have over here. And then we have a second decision, which is the one in which I would like to focus, in which the purple cells become either primitive endoderm or epiblast, and the epiblast cells are becoming the embryo itself. And these are the ones that I think Silvia was talking about when she was referring to naive pluripotent cells yesterday. So we are interested in this second decision. Why? Because Kat and Nestor have a nice way, and which is not unique. I mean, other groups have it, but they were studying this kind of a cellular decision-making process at this stage. And this is an example of the type of data that they have. So here you have an embryo which has been stained and fixed over different stages. So we are going to measure time in this talk, at least at the experimental level, in terms of the number of cells that the embryo has. And this is basically a percentage of what is the percentage of cells in the ICM that become either still non-differentiated or they differentiate into primitive endoderm or epiblast. So here you can see in the color code, the purple, red, and blue, these are the ones that we are going to be interested in. And you can see here, every one of these columns corresponds to one embryo with one given size. Of course, this is not linearly distributed along this axis because they don't have one embryo exactly at each at each cell number. But this gives you more or less an idea of what happens as the embryo grows. And what we can see at the beginning, most of the cells are undifferentiated in the inner cell mass, and eventually the cells differentiate into these two fates, the blue fate, primitive endoderm, and the red fate, epiblast. And in average, what we can see in a very robust manner is that the differentiation is quite symmetric. It's almost 50-50, not exactly. The gray cells here are basically red cells. This is just a matter of how they are classified and what happens downstream after the decision has been made. But at the end of the day, what happens after this stage around 150 cells is that we have a very nice balanced decision, almost around 50-50, at least for a theoretician's, from a theoretician's point of view. So the question is, what kind of network gives rise to such a cell-fate decision, which is rather symmetric? And of course, we have answers from the point of view of theory and modeling. And one answer was provided around 15 years ago already by Sui Huang and collaborators when they were studying lineage commitment in the hematopoietic system. So these were already adult cells. But there you also have the same problem. You have the problem of differentiating into two fates in a more or less balanced manner. And you can get this by assuming this kind of mutual inhibition circuits, either by themselves or with the inclusion of a direct positive feedback among the two key regulators here. Okay, so these type of circuits are going to give rise to a situation like this in phase space. In here we have the two key regulators, the concentrations, and we can see that we can go to either one or to the other fate. And more or less the areas of phase space that go to one or the other are more or less balanced, and depending on how balanced they are, we can get a balanced cell-fate decision. So this means that we have already, if you will, a type of circuit that could be associated with this type of behavior. And indeed, if you look at if you go back to the embryonic cell-fate decision that we were discussing before, we have already candidates for these key regulators. In particular, this would be GATA6 for the primitive endoderm blue fate, and NANOC for the epiblast red fate. And the usual assumption made in the literature is that these two factors inhibit each other, and maybe they also self-activate. Now, I have to say that, as Nadine was showing earlier, of course, the regulation of transcription in mammalian cells is very complicated. So, I mean, it's hard to really have direct evidence of this kind of interaction. So most of the evidence that we have of these interactions is indirect on the basis of the effect of mutants or different types of treatments, what happens in this case, and we can infer what is the type of interaction that we have here. So I think it's safe to say that these interactions are considered effective in some sense, because we have no direct evidence in many cases. However, I mean, this has been established as already common knowledge that this could be underlying this kind of cell-fate decision. So the question would be, whether this decision is cell-autonomous, because what we could imagine is that every one of these cells in here is throwing a coin in some sense and decided whether to go to the blue or to the red fate in an autonomous manner. So the question is whether this is what happens, and then we get just 50-50 because the chance of the cells going into one or to the other fate is 50-50, and that would be enough to produce a reasonable outcome at the average level. So the question is whether this decision is cell-autonomous. So the first thing I wanted to show you is an experiment that Nestor did to address this question, whether this decision could be cell-autonomous, okay? So let me go through it relatively quickly. The idea is that they mix two mice. One of the mouse line is basically a mutant for GATA6, which means that these cells here cannot be primitive endoderm. They can only be, sorry for the color here. This is not red, but anyway, I hope you agree with me. These cells cannot become primitive endoderm, so they are forced to become epiblast. Okay, so this is one of the mice, and they cross this mouse with a mouse which is considered wild type. We just have a GFP so that they can be these cells can be identified, but in all regards, these cells can become either epiblast or primitive endo, and the green ones, okay? These are the ones, the red ones or the pink ones are the ones that are restricted to be epiblast. So what they did is, what Nestor did was reaggregation experiments in which they were combining the two types of cells, or they were generating embryos from these mice, disaggregating the cells and reaggregating them, and then varying the percentage of each one of the two cell types. So we can go from a mouse in which all the cells were forced to be epiblast to a mouse in which most of the cells, or eventually all of the cells, could choose their fate, as in a normal wild type mouse, okay? So the question is, in each one of these cases, what was the decision being made by the cells that can make the decision, which are the green cells? The green cells are the cells that are allowed to make the decision. So what decision was being made by the green cells in the presence of increasing amounts of red cells, of red forced cells? So first of all, let me remind you, this is the typical wild type scenario in which, as I was telling you earlier, the decision is rather balanced, almost 50-50. And this is what happens. This is the fate of the cell, of the red cells, of these cells here, the red forced cells. This is their fate. And this is the fate of the normal cells from the green mouse. And as you can see, of course, the cells that can only become epiblasts, they will become epiblasts, trivially. But the cells that they can choose, then they choose to be almost in balanced situation. If, let me tell you here that this axis indicates what is the percentage of green cells in the embryo. So this area, this point here is basically close to the wild type. Almost all of the cells in this embryo are green. So this means that there are not too many that are forced to be red. Those that are forced to be red, of course, are becoming red. And the ones that can choose, they mostly choose 50-50. There is a slightly imbalance here, which is already very telling, that it's not as much as here. So some of the cells seem to be, so there's a slight preference of becoming blue, because there are some deficit in blue cells in principle. And indeed, if we go to a situation in which more and more of the cells in the reaggregated embryo are forced to be red, what happens is that the cells that can choose, they choose to become mostly blue, which means that this decision is not cell autonomous, it depends on the neighbors, okay? So I think it is safe to claim that this decision cannot be cell autonomous, because depending on the neighbors that the green cells have, they're going to decide in one way or another what to do, okay? So there has to be cell-cell communication going on and governing this decision. So now, how do we go from this model, which is basically cell autonomous, to something that involves cell-cell communication? So basically, we can go back to the literature, and one possibility, which is the one that we ended up considering, is one in which cells communicate with each other via FGF signaling, and FGF was already discussed earlier, again by Sylvia, if I remember correctly. And the idea is, okay, now we have a little bit this somewhat complicated situation in which we have our two key regulators, which are talking with each other between different cells via FGF. Okay, and indeed we can double check again using mutants that FGF is necessary for this balance decision, because if we knock out, Nestor and Kat did knock out FGF, we can knock out either FGF production or the receptors, because we were considering two receptors here, FGFR1 and FGFR2. I'm not going to get into details and into the difference of these two receptors right now, because that's not the topic of the talk. But if we knock out the ligand, the signal, or we knock out the receptor, in both cases, basically, as you can see, the embryo cannot make a balanced choice. All the cells go to the red fate. Here, basically, this is the double knockout. There is no receptor whatsoever, or there is no signal, no ligand whatsoever, okay? So basically, you have that the decision cannot be made. So you do need FGF for the decision to happen. So that's the first thing I wanted to tell you. So then, so far, what do we have? So far, I think we can conclude that intercellular signaling, cell-cell signaling is necessary for the decision. But then what I wanted to know was whether it is sufficient, okay? So is it just something that happens on top of whatever else that we were assuming, or is it really sufficient cell-cell signaling for the decision to happen? Okay, and for that we wanted to, the best way to try to answer this question is using mathematical models. And for that we were inspired by a very beautiful paper that Genevieve and Didier and co-workers in the Brussels group published a few years ago. It was a very nice work in which they have basically all the components. They had the direct mutual inhibition and signaling, And we were analyzing this model for a while, but eventually we got to the conclusion that if we wanted to answer the question about sufficiency, about whether the cell-cell mechanism is enough, then we needed to somehow disassemble or separate the two effects, the mutual inhibition from the signaling. And it was hard to do from this model because it was very easy to lose the right regime of a balanced decision, which they nicely managed to get. So what we decided to do eventually was to try to simplify the model. And the idea, this is our version of the model that we were just discussing, where we have both the direct cell-cell autonomous interaction here between GATA6 and NANOC and the cell-cell communication route. And then what we try to do is we try to simplify. So what we did is, okay, let's forget for the moment that we don't have a mutual inhibition. Let's forget also the direct feedbacks that maybe they are not necessary. So again, we are looking for simplicity. And then we transform this model into this simplified version, okay? So now we are going to consider this simplified version in which NANOC and GATA6 are interacting via FGF. Now, in this model, if we look at it closely, we can see that, for instance, there is a path going from GATA6 to NANOC, which is an inhibitory path. It is mediated by FGF signaling, of course, and ERK, which we haven't mentioned, but of course this is mediated by ERK signaling, but it's an inhibition from GATA to NANOC. So we could effectively draw it in a way like this. So we have this inhibition. And as you can imagine, then we can go in this other direction. And we also have an inhibitor interaction from NANOC and from NANOC to GATA, which corresponds to this other situation. Which means that now we have this system that we are going to consider right now, this circuit, is basically equivalent to a mutual inhibition circuit that is mediated by the signaling. So it's no longer cell autonomous. And the nice thing about this model is that it's relatively simple, so we can model it. And again, there are several assumptions made here. We ignore explicitly the dynamics of ERK because we already assume that it's in quasi-equilibrium, if you will. So at the end of the day, what we have is we have this system of equations for these three players, NANOC, GATA6, and FGF. Okay, and now what we have is that the role of cell-to-cell signaling is played by these production terms or disactivation terms of NANOC and GATA, which involve FGF average over the neighbors. So these terms here correspond to the coupling between cells, so they are going to be key in the operation of this model. Okay, so now the other thing that we can do, I mean, after all, we have already eliminated the dynamics of ERK, so we could as well try to make this model even simpler to try to see whether we can get some understanding of how the model is going to behave. So we can assume that 2 of the 3 degrees of freedom are again in quasi-steady state, for instance, GATA and FGF, and this allows us to eliminate these 2 equations, and we eventually end up with a single equation for NANOC, which we call now X because it's a dimensionless version. Okay, so that's the equation that we have. So this means that every one of the cells in the embryo is going to be represented by this equation. This equation tells us what is the dynamics of NANOC, and from that we can obtain the dynamics of GATA6 and FGF4. Okay, so let me just so this is just a summary of the model as it is right now. This is a dimensionless model. It's a model that determines what is the level of NANOC in each one of the cells, and the coupling is via the average over neighbors, which is down here. Okay, and also interestingly, since this is a dimensionless model, we basically have only 2 dimensionless parameters: this alpha here and this k. Okay, and we can put in terms of all the other parameters of the more complex model, if you will. So we have these 2 parameters and the corresponding Hill functions. But now the advantage is that this model is relatively simple, so we can start analyzing it. Okay. Now, of course, the problem is that this is a model for n cells. So the first thing what we could do is we could simplify things and analyze what happens if we only have 2 cells. Of course, this is a completely imagined situation, an idealized situation in which we only have 2 cells. So if we only have 2 cells, this means that this is our system. Okay, so we have equations for cell A and for cell B. They are coupled with each other through this term here, and basically we can analyze the dynamics of the system by plotting its behavior in the phase plane formed by XA and XB, and these are similar plots to the ones that I was showing you earlier, given by Sui Huang. Okay, and what you can see in this case is that these are the null clines, by the way, the 2 null clines for those of you who are interested in this type of thing. The red and the blue are the two null points of the system. And what happens is that these two null points cross in these two stable fixed points. This is an unstable fixed point. I'm not even highlighting here. So basically, this supports the idea that the system can be in one of two states. Either cell A is high in NANOC and low in GATA6, or cell B is high in NANOC and low in GATA6. Okay, so these are the two states of the system. So now we have simplified the model to the most minimal description, and this might remind you, maybe some of you, to another type of cell-cell communication system in which we have bistability, which is the case of lateral inhibition. Okay, so in this particular case, we have from a conceptual point of view, what we claim is that the system behaves as if it was a lateral inhibition model. At the end of the day, we assume that the FGF is not diffusing far away in the early embryo. From the conceptual point of view, we assimilate our model to a lateral inhibition model, in which, of course, we have two types of cells which are next to each other. So this means that we have two cell types coexisting in a population. Of course, now we are not claiming that we have a spatially distributed population. In the early embryo, the cells at the beginning are mixed together. I will show you some simulations in a minute. But the idea is, okay, now we have two populations of cells. In principle, such a model should be able to reproduce a system in which cells are clustered into two states. We could call them cluster A and cluster B. So now this means that we can interpret our model, the same model that I was telling you earlier, that we can draw in phase plane, like this, with our null lines. Now we can interpret it as an ansatz for two populations. So we can consider that we have two populations that we call A and B. So this means that this is the level of NANOC in cells of type A and the level of NANOC in cells of type B. And as before, basically what we have is two states separated by this boundary, and then we have these two basins of attraction. And this means that we have the two situations in which the cells can either be high in NANOC and low in GATA6, or vice versa. Okay. So now the idea is, okay, from the conceptual point of view, this model seems to explain, or should be able to reproduce this kind of almost 50-50 split that we see experimentally. And also, the idea is that this 50-50 split is an attractor of the dynamics. So the embryo starts somewhere in here, and eventually it's being attracted to these two states that correspond to the two fates. And this is basically what, again, this is just a reminder that indeed in the experiments, we do see that this balanced state is an attractor, if you will, of the dynamics, and we can quantify it better. These are different quantifications that we did using the study that in which we were working on with Kat and Nestor, previous work by the same group, and we always see that at the beginning we have large variability in the ratio between primitive endoderm and epiblast cells here, and eventually this variability is reduced up to a situation in which more or less this is a balanced situation in which this logarithm is close to zero. Okay, it's not exactly this, there's a little bit more of primitive endoderm cells, but again, this is basically, we can interpret this as an attractor, if you will. Okay, so now, of course, this is still very conceptual. How do we test our model? How do we test that the model behaves in the way that it should? So what we did is we went back to the paper that I was telling you earlier by Genevieve and Didier, and they also had in that paper a very nice way of modeling the embryo from a mechanical point of view. So basically, we implemented their mechanical model in which basically the cells were interacting via this type of force, which had a repulsive component, of course, and an attractive component. And this was enough for them to reproduce the mechanics, if you will, apart from the biochemistry of the model. So we implemented this type of model, and we put on this model, on top of this model, we put the simple, minimal biochemical model that I was telling you earlier. So, and this is just an example in which if you run the simulations, you can just see that the cells at the beginning are purple, and eventually they're going to start differentiated into red and blue cells. And eventually what we end up having, ideally, that's what we want, I want to show you, we end up having a 50-50 split, which goes beyond the simple phase plane two-dimensional description I was telling you earlier, okay? So, and this is an example of the quantification. That's what we do when we apply the model that I was telling you a minute ago. Then we quantify, also including errors, and we see that in a time frame which is similar to the one that they see experimentally, the purple cells are losing their are basically deciding to go into red or blue, and eventually we have an almost 50-50 split, slightly biased in favor of the primitive endoderm cells, in consonance with what Nestor and Kat observed and also in previous studies, okay? So in principle, this type of model is working not only on paper or not only at the level of the phase plane, but also when we implement it on top of a mechanical model in which cells are moving and they are proliferating, of course, and their neighbors are changing, the neighbors with which they are exchanging FGF with, they are changing, and still we get something that seems to be robust, but of course that's what we need to test now. So so then the next thing that we did is, okay, let's see if we can recapitulate several of the observations that they made. And for instance, the first one that we looked at is an observation regarding scaling. This is data that they had published a few years ago. Nestor and Kat did. And what they did in that case is they took an embryo at the 8 cell stage, and they saw that if you split it in two, then you end up having two embryos which have, again, the right proportion once more of red and blue cells. So here you have it here. This would be the half. This is the control case, and this is the half embryo, and you still have the 50-50 split, more or less 50-50 split. And also when they aggregate two embryos and they build a 2x embryo, larger, again, the system self-organizes and produces a 50-50 split, okay? So this was their observation. So the first thing that we tried to see whether our simple, very simple model was able to recapitulate this, again, without any need of a cell autonomous switch. It's all driven by FGF signaling. And indeed, that's what we did. I mean, let me remind you that we don't have parameters to tune only alpha and K. We didn't have to tune anything. As soon as we get the right cell-fate decision profile in the wild type, we can go to half the embryo or 2 times the embryo, and we still get a nice 50-50 split in our model, okay? And so I have to tell you, the decision happens at the salt and pepper pattern. I'm not going to enter into the dynamics. We also have a segregation of the two fates, but this doesn't seem to be necessary because just the mixing of the two fates and the FGF signaling is enough to self-organize the system into this more or less 50-50 split. Okay, so that's the first thing that we just looked to check that the model was minimally reasonable. The next thing that we wanted to check, and that's what I'm going to be talking about in the next few minutes, is how robust is the attractor? Is this attractor when in proliferating embryos? So it's not only, I mean, the perturbation I was just showing you earlier, it was big because it was either halving or doubling the embryo, but it was still, maybe one could imagine that, oh, maybe everything renormalizes and in average the decision happens. But what happens if we perturb the embryo in a more subtle manner, if you will? So that's what we did next. And we used the ability of Nestor and Kat in perturbing these embryos. And let me tell you about 2 perturbations that they did. The first perturbation that they tried was adding mouse embryonic stem cells to the embryo at different stages, okay? Now I have to tell you, these embryonic stem cells are basically coming from an epiblast. So they are cells that are in principle of the red fate, but they are already much smaller because these cells have already spent time in this fate. And as you probably have seen from the movies, the cells are dividing and they are not changing in volume. So that's why they're becoming smaller as the simulation proceeds. And that's why we have, this is the idea. The idea is that they add these smaller cells that are committed to becoming red cells. So basically, I just labeled them, or we just labeled them as this kind of yellowish, greenish color, but at the end of the day, they are basically committed to becoming red. Okay, so and then we can change the number of cells, and we can change the time at which these things are added, and in that way we are forcing the decision once more. Okay, by exogenous cells, and we can model this in our experiment, in our simulations, of course, very simply, very fast, because the model is very simple. So we can add the embryonic stem cells at any point. At some point, the network, we turn it on, because at the beginning the circuit is off, because we consider that the levels of signaling are not enough for epi and for the NANOC and Agata6 to interact. But anyway, we can change the timing at which we add the embryonic stem cells, and then we can see what is the final decision. And this is the outcome of the model, basically, more or less in the same kind of representation that you have seen earlier. So basically here, This is going to be, these are the embryos labeled or ordered in terms of the final number of ESCs, okay? So this is the final number of years of embryonic stem cells that they have after 48 hours. Let me, of course, emphasize that these cells are dividing, and the only thing that they can do experimentally, and that's why we did this in the model. We can quantify this the timing of perturbation and the intensity of the perturbation in many ways, but we try to mimic what they do in the experiment, which is they know at the end of the experiment how many ESCs they have. So basically what happens is that here in this region, we have added a lot of embryonic stem cells, okay, because now there are a large fraction of the cells in the ICM are basically embryonic stem cells, whereas in this area here, there are not so many. And what you can see, interestingly, is that there is a region, quite a large region, in which we have added embryonic stem cells up to a certain point, okay? And if we don't add too many embryonic stem cells, what happens is that there is a robust response in the sense that as we increase the number of embryonic stem cells that are being added, there are less and less of the native cells of the embryo that are becoming red, because the cell doesn't need so many red cells. So basically, this keeps the ratio between blue cells and red plus yellow or red plus green cells more or less constant, and this indicates a robust response. This means that the embryo would be viable in this condition because the split between the two fates is more or less constant, even though we have been increasing the number of embryos of stem cells that we have been including. Of course, eventually, when we are here, the system is basically overrun because we are adding so many embryonic stem cells that we have a lot of red cells. And eventually the native cells cannot keep up, and you don't have enough primitive endoderm cells at this point. So these are not viable embryos, but there is a region in which the response is rather robust. So they did this is just the model. So they did the experiments, and here you can see the result. Here you can see that there is a large region, and here, maybe this is clearer here. This is basically the number of ESCs that they added as a fraction of the number of epiblast committed cells at that point. And what you can see is that in this wide region, the response is robust in the sense that we basically end up with a 50-50 split between primitive endoderm cells and the rest. Okay, so this is basically control that we should not compare this here because this is just a representation of normal cells that have been normal embryos that have been untreated. Okay. But here you can see that there is a region of robust behavior, and then here the system is overrun. So qualitatively, our simple model also reproduces the type of observation that we have here, meaning that we have a robust response to a perturbation in the form of an expansion of this epiblast pool, which is driven by the addition of exogenous external embryonic stem cells. Okay, that's perturbation number one. And finally, and I think that I am going too fast, but anyway, we will have a lot of time for discussion. The second perturbation that they could do also is they could eliminate cells of different types. Okay, so the idea is that first of all, they mix basically wild type mice, but they just have some markers. Okay, so if you have a mouse here in which we have an ubiquitous nuclear marker, so that we can tell where the cells are, and then here we have a marker. A mouse which we are going to label in green, and this green doesn't mean the same as the green in the previous slides, some slides ago, so I apologize for it. But this green mouse here means that the cells coming from these mice are basically, if you see them green, this means that they are already primitive endoderm specific. So in that way, we can distinguish between cells that are in the primitive endoderm fate, which we were labeling blue before, but here they are basically going to be labeled as green. This is just because we were using GFP. So we're going to have cells that are in the blue fate, which here are represented as green, and cells that are in that are going to be deciding. In principle, they are going to be, if they are not green, this means that they are going to be end up in the red fate. Okay, so we can just see what type of cells we can address, and then what they can do experimentally is they can ablate the cells by using laser ablation. Okay. And this ablation can be targeted of random because you can choose which cells you kill. Okay, so the nice thing is that they can kill cells at will, and they can kill cells of one type or the other depending on their color, or they can just do it randomly. So the idea is, okay, they take the mouse as it's growing, the embryo, they kill the cells and then let it grow, and then they fix, and then they stain it. And then you can see what is the fraction again of cells in one phase or the other. So that is the experiment. And before I tell you the results, let me show you what the model is if you will, predicting. And this is, I'm going to now show you a slide with a lot of data. So I'm going to be going slowly since I have some time. So first of all, this is what happens if we kill the cells randomly. In this particular case, we kill the cells randomly in silico in the model. And here we kill up to 30% of the ICM. So of course we can follow single cells. We can look at what happens at the average level. Okay, and you can see here that the total number of cells is lost. At some point we lose cells simply because we are killing them randomly. Okay, this is not too much to perturb what happens in the different fates, but we still, in the total number, we see this thing going down. And what happens is that both in the 30% reduction case, which is this one here, both in this case, or of course, in the case in which we kill much less cells, if we kill them randomly, the system is very robust, okay? At a wide time window, okay? So at any time that we perform this ablation, the embryo is able to recover, and eventually you have an almost 50-50 split, okay? So this means that, again, the embryo is robust to a perturbation in which we remove cells randomly. Now, what happens when we remove cells in a targeted manner? So here we have, on the middle row, we have reduction of primitive endoderm cells. And again, well, in these simulations here, I am showing you the case in which all the primitive endoderm cells are removed, all of them, 100% of the cells are removed, okay? Or 100% of the epiblast cells are removed. And they are removed at different times. So this corresponds to the different times at which the removal takes place. And what we can see is that if we remove only 30%, the embryo is quite robust. It's still able to recover, okay? So it's losing 30% of the cells at different times. And as time moves on, let me remind you that the number of progenitor cells is going down. Eventually, of course, this would have a limit. But since it's only 30% of the cells that are being reduced, the system is quite robust up until this number of cells at the time of perturbation. And what we can see here is that if we remove 100% of the primitive endoderm cells, at some point, when the removal happens at late stages, then this means that the embryo doesn't have time. So we have removed basically all of the primitive endoderm cells, and then when we wait and see what happens, eventually the progenitors are basically disappearing and they cannot repopulate the blue cells, and eventually these embryos are not going to be viable. Okay, so this indicates that there is a limit in the robustness, but the interesting thing is that the embryo is relatively robust for a wide range of perturbations. And the same thing for the epi case, okay? If we kill 100% or, well, 30, if we kill 30% of the cells, the embryo is quite robust. It's always able to recover, at least at these levels, at these times of removal. And if we remove 100% of the cells, eventually the system cannot recover because the embryo doesn't have enough time to repopulate the epiblast pool, and basically most of the cells are going to be primitive endoderm. This means that these embryos are not going to be viable. Okay, so anyway, yes.
If you could conclude quite fast, because
yes. Oh, thank you. Sorry, I'm already. I was kind of going slow, but anyway, I'm done. Anyway,
that's fine. That's fine.
I'm just going to show you the experiments. And these are the experiments. So these are basically the results by when either primitive endoderm or epiblast cells are removed or randomly. You can see that randomly there is quite a bit of robustness, but I would like to direct your attention to these cases. This is a 100% removal, and we can see that if the removal takes place at early times, we still have a nice 50-50 split, but eventually, if we remove too many primitive endoderm cells, the epiblast cells take over, or vice versa. So again, this is showing in a qualitative manner that there are limits to the robustness, but if the perturbation is not too strong, the system still can recover. So anyway, that's all. So I want to summarize what I have been telling you. Point number one is that I think we have the feeling that our results support that cell signaling via FGF is both necessary and sufficient for cell-fate decision in the early mouse embryo, that the mechanism is somewhat analogous to lateral inhibition, also at the conceptual level, and the decision is robust to tissue site perturbations. So I would just like to thank again Nestor and Kat and Laura, and thank you so much for for your attention.
Thanks a lot for this very clear talk and impressive results, really. So we have time for a few questions. So Thierry Morin.
Hi, thanks for the great talk, very interesting.
Thank you.
I just had two quick questions. The first one is, do you assume mean field, or is they really set, you know, for the signaling, or is it just neighbors that communicate with each other? And the second question is, can you say something about this segregation that you didn't talk about? Do we understand the mechanisms by which these two types of cells then segregate?
Yeah. Okay. So first of all, regarding mean field, we do not assume mean field. We could but we just biologically, and maybe you biologists in the audience can correct me if I'm wrong. Our assumption is that FGF is not going to be traveling very far in these embryos at this stage. But still, I mean, what I can tell you is that if we increase the range of the coupling, we still get the 50-50 split. But we put ourselves in the worst situation in which there is no mean field. So that's the first thing. So if there is just nearest neighbor coupling, we still have a very robust 50-50 split. And the other question was the segregation. So what we assume is something that is pretty standard. I think Genevieve and Didier and Alex were also assuming this in their model, which is that and again, I'm not sure if there is a lot of evidence about this, which is that the addition between pairs of cells of the same type is different from the addition of pairs of cells of different type. And just adding this, which we did add, adding this was enough to lead to a segregation. So we went from a salt and pepper pattern to a self-segregated pattern. Of course, we were not very much worried about the specific geometry of the blastocyst at the late stages, but we do get the segregation just by assuming differential addition. So that's the. Thank you.
Okay, Didier.
Yes. Yeah, thank you for this nice talk. I would be curious to see further the differences between your framework and ours. But maybe one question. So each cell has the same equation, the same kinetics. So I was wondering about the initial heterogeneity. So this is what we discussed with Genevieve many times. Did you include variability on the initial conditions? Or cell division, or is cell division enough to create some inhomogeneity in the system?
Okay, we include both things. I don't think we need a large heterogeneity in the initial conditions, but we included both heterogeneity in the initial conditions and then variability in the timing of cell division. This is something that we also try to match with the experimental observation. So we had we had a relatively good understanding of the variability in cell division from the experiments of Nestor, and we try to match this variability in our model. So it is true that our model includes both variability in cell division and variability in the initial condition of the cells. But I think neither of them are absolutely necessary to get to the segregation. Both of them will be enough as sources of noise to get to a 50-50 split, I think.
Okay, and
thank you.
Thanks.
A last quick question from Peter.
Yeah, thanks, Yuri, for the very nice talk. I just have a question of why is it that if you have mutual inhibition with basically the cell-cell signaling that reinforces this, that you do not end up with a situation where you only have cells of type X or cells of type Y only? So what controls this 50-50 ratio?
Yeah, okay. One thing, I mean, we were talking with Didier about the initial conditions. So it is, I agree with you, it is important to take into account what are the initial conditions in the system. And an important point that it's observing experimentally is that the cells start with this in this kind of double positive state, which means that they have both high levels of both nanoc and primitive endoderm. I agree with you, Peter Ryan, that if we don't include this kind of symmetry, then the cell fate decision maybe would not be so symmetric, okay? So it is important to take into account that this circuit is turned on at a time in which these cells are in this double positive fate.
Okay. Well,
but that's all you need, and I think it's safe to make this assumption because that's what is observed experimentally. Thank you.
Thanks.
Okay.